Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Genomics Explained: Structural and Functional Insights. Table of Contents. Introduction to Genomics: Structural and Functional. Categories of Genomics. Methods in Structural Genomics. Methods in Functional Genomics. Applications of Genomics. Introduction to Genomics: Structural and Functional. Genomics is a scientific field that focuses on studying the complete set of genetic material, known as the genome, present in an organism. It involves examining the structure, function, and interactions of all genes, along with other components that constitute the genome. The genome consists of DNA, which holds the biological instructions that determine how an organism develops, operates, and interacts with its environment. Through genomic studies, researchers gain insights into an organism's characteristics, behaviors, and its susceptibility or resistance to various diseases. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Categories of Genomics. Genomic studies are broadly divided into two main categories. 1. Structural genomics. 2. Functional genomics. Structural genomics focuses on understanding the structure of genome sequences. It includes constructing genome maps, sequencing genes, annotating gene features, and comparing genome structures to gain insight into the organization of the genome. Functional genomics focuses on studying gene expression and determining the functions of genes within a genome. It involves analyzing gene activity on a genome-wide scale, using high-throughput techniques to understand how genes interact and contribute to biological processes. Beyond these two main branches, there are several additional subfields of genomics that explore specific aspects of genetic research. Epigenomics studies epigenetic modifications, collectively known as the epigenome, which are chemical compounds that attach to DNA and regulate its activity without altering the DNA sequence. These modifications, such as DNA methylation and histone modification, play key roles in controlling gene expression and contribute to differences between various cell types in the body. Metagenomics investigates the genetic material obtained from entire biological communities rather than individual organisms. It is mainly applied to microorganisms and helps study the diversity, composition, and functional roles of microbial communities in environments like the human gut, soil, or ocean. Pharmacogenomics uses genetic information to guide and personalize medical treatment by determining the most effective drug types and dosages for individuals. It helps predict drug responses or toxicities and supports the development of personalized medicine. Comparative genomics involves comparing genomes of different species to understand evolutionary relationships, genetic variations, and conserved functional elements. It employs computational tools to identify similarities and differences between species genomes, providing insights into their evolutionary history and biological functions. Download our microbiology study app made for researchers like you. Methods in structural genomics. Genome mapping. Genome mapping is a key step in structural genomics that focuses on identifying the specific locations of genes, mutations, or traits on chromosomes. Different types of genome maps are used to represent varying levels of detail. Genetic linkage maps. Genetic maps are low-resolution maps that determine the relative positions of genetic markers based on inheritance patterns. Physical maps are high-resolution maps that locate recognizable landmarks on genomic DNA independently of inheritance patterns. Cytologic maps, chromosome maps, describe visible banding patterns on stained chromosomes, which can be observed under a microscope and serve as chromosomal markers. Genome sequencing. Genome sequencing provides the most detailed genome information by determining the complete DNA sequence of an organism's genome. One of the most widely used DNA sequencing techniques is Sanger sequencing, which uses fluorescently labeled dideoxynucleotides to terminate DNA chains at various lengths. The resulting fragments are separated by electrophoresis, and the sequence is determined by interpreting the banding pattern or chromatogram using base calling, where each nucleotide is assigned based on the peaks. Whole genome sequencing can be performed using two main approaches. Shotgun sequencing, where DNA is randomly fragmented, sequenced from both ends, and then assembled computationally to reconstruct the complete genome. Hierarchical sequencing, where larger DNA fragments are first mapped using physical mapping techniques, cloned into bacterial vectors, and then sequenced in an ordered manner to determine their position and order on chromosomes. Genome sequence assembly. Genome sequence assembly is the process of reconstructing the entire genome from many short DNA fragments known as reads. Sequencing reactions generate short reads that must be joined together by aligning overlapping regions to form continuous sequences. The first step, base calling, determines the nucleotide sequence of each fragment and assigns a quality score to represent the accuracy of each base call. The next step is assembling the short reads into longer sequences called contigs by identifying overlaps, determining their order, and deriving a consensus sequence for each contig. Common software used for sequence assembly includes FRED, FRAP, TGR Assembler, and Arachne, which process raw sequencing data to generate assembled contigs. Genome annotation. Genome annotation follows genome assembly and involves analyzing and interpreting the sequence to identify biologically meaningful features. The annotation process includes two main steps. In gene prediction, computational tools are used to locate and define the structure of genes within the genome. In functional assignment, predicted genes are compared with known genes using homology searching methods, such as BLAST, to determine their possible biological functions. The annotated data are stored in public databases like GenBank, where they are made available for researchers to conduct further studies and comparative analyses. Methods in functional genomics. Genetic interaction mapping. Genetic interaction mapping is a method used to explore the functional relationships between genes by studying how multiple genes interact to influence specific phenotypic traits. This technique involves pairwise gene perturbations, such as gene knockout, knockdown, or overexpression, to observe how the modification of one gene affects the phenotype produced by another. One key form of genetic interaction is epistasis, where the effect of one gene is masked or modified by the presence of another non-allelic gene. Various computational tools are available to detect and analyze gene-gene interactions, including BEAM, Bayesian Epistasis Association Mapping, TEAM, Tree-Based Epistasis Association Mapping, BOOST, Boolean Operation-Based Screening and Testing, and TSGSIS, Two-Stage Grouped Sure Independence Screening. Genetic interaction mapping is valuable for discovering new gene functions, understanding gene networks, and organizing gene products into functional pathways and complexes within a hierarchical framework. Microarray technology. Microarray technology is widely employed in functional genomics for global gene expression profiling. A microarray consists of a chip with a high-density array of immobilized DNA oligomers or complementary DNAs, each serving as a probe that binds to a specific complementary sequence. In a typical microarray experiment, fluorescently or radioactively labeled cDNA molecules are hybridized to the probes on the chip, and the intensity of fluorescence or radioactivity at each spot indicates the level of mRNA expression in the cell. This approach allows for the systematic analysis of gene expression patterns, facilitating the identification of novel gene functions and regulatory mechanisms. Microarrays have several important applications, including comparing gene expression between normal and diseased tissues, detecting single nucleotide polymorphisms, SNPs, and identifying changes in chromosomal copy numbers. SAGE, Serial Analysis of Gene Expression, is a high-throughput, sequence-based technique used to analyze and quantify global gene expression levels. Unlike microarrays that depend on hybridization, SAGE relies on direct sequencing of RNA molecules, providing a more comprehensive and unbiased analysis of gene expression. The process involves isolating short sequence tags, SAGE tags, from mRNAs, which serve as unique identifiers for specific gene transcripts. These tags are then concatenated, cloned, and sequenced, allowing for computational analysis where the frequency of each tag corresponds to the level of gene expression. Although SAGE provides detailed and quantitative insights, it has certain limitations, including high cost, time-intensive sequencing, and difficulty in determining the optimal number of tags needed for complete transcriptome coverage. Applications of genomics. Genomics plays an important role in the diagnosis and classification of diseases by identifying specific genetic variations or mutations associated with particular conditions. It enables researchers and clinicians to study the genetic basis of diseases, identify genetic markers linked to specific disorders, predict disease susceptibility or risk, and design targeted therapeutic approaches for more effective treatments. A major application within this field is pharmacogenomics, which examines how genetic differences influence an individual's response to drugs. It helps determine the most effective and safest medications for each person, paving the way for personalized medicine tailored to individual genetic profiles. Comparative genomics involves comparing the genomes of different species to identify genetic similarities, differences, and evolutionary relationships. This comparison enhances understanding of trait development, evolutionary processes, and the genetic diversity of life on Earth. In agriculture, genomics contributes to improving crop and livestock breeding, increasing disease resistance, and boosting agricultural productivity. It helps identify genes responsible for desirable traits, leading to the development of superior crop varieties and animal breeds, with enhanced performance and resilience. Genomic analysis also finds significant use in forensic science and criminal investigations. DNA evidence obtained from crime scenes can be analyzed to identify suspects, confirm biological relationships, and establish connections between individuals and evidence, thereby supporting justice and law enforcement efforts. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!